this is a meeting about innovation. People ask me all the time, why did you come to Pittsburgh? Why did you stay? I interviewed at Pittsburgh because one of my mentors at Vanderbilt was Dr. Tarpley, who was a general surgeon who was good friends with Leland Albright when they were interns, I believe. And I came up here because he told me I had to, and I absolutely fell in love with the place. So I was happy I ended up here. But what keeps me coming back is the environment of innovation. And so I'm going to talk about where Pittsburgh is in the world of brain-computer interface and leave you with some thoughts about where we may go. There are a number of devices here that are not FDA approved. They are all being used under investigational device exemptions. So I'm going to see how this works because I don't have a space button, so let's see. So I'm going to talk a little bit about the different ways we can do brain-computer interfaces. And so if we start with non-invasive techniques, we can do EEG and FMRI and MEG. And so there's lots of toys you can buy out on the market that are supposedly EEG devices so that you can levitate objects. So people like these ideas because they are non-invasive, but they probably don't give you a whole lot of degrees of freedom or things that you can do. So the next thing that we can do are these electrodes that would sit on the surface of the brain. So this is more invasive, but doesn't actually penetrate the brain. They can produce very stable recordings. And in fact, it sounds kind of strange, but one of the nice things is they limit the amount of data you collect, so you don't have to figure out how to process giant amounts of data. If we then go to electrodes that actually penetrate into the brain, we have a wealth of data that we can collect. And the thought is that it should allow you for more intuitive control. And some of the limitations are the fact that it is invasive, and of course you have this vast amount of data you have to figure out how to handle. So if we look at the non-invasive techniques, a lot of people have looked at FMRI and MEG. These are big, giant, clunky machines, so they're probably not going to be particularly useful as a take-home brain-computer interface, but they can be very helpful from the standpoint of mapping. So we use a lot of fMRI mapping, and we're primarily working with patients that are quadriplegic, paraplegic, or who have loss of their upper extremity function. And so while most of you think of fMRI as someone laying in an MRI scanner performing a task, a lot of our patients are actually imagining a task or watching video cues. And we know from the vast amount of work that's been done on spinal cord injured patients, you still get brain activation even when you can't move the limb. We can also use the MEG. Again, we do the same sorts of things where they're imagining the task or they are watching video cues. And one of the things that we do, and one of the things that's actually part of our screening, is we would like to see correlation between the FMR activation and the MEG activation, because we feel that that helps us better target the active areas. It also turns out that you can do at least some kinds of therapy using MEG, and this is an example of a study that was being done at the VA. And so it's an MEG study, and so the MEG responses are being recorded, and then they are sorted. And what the subject is doing is trying to open and close their hand, and at the same time, they're watching a video of a hand opening and closing. And so the idea behind, and then they get feedback, and so this then changes the gain. So as they try to open and close their hand, they get a little bit more visualization on the video. And the thought is that with long term, this might actually work to enhance circuits to improve. But these are really proof of principle studies, and so right now we are working on setting up some long-term studies, particularly in the VA population, to look at this. Could you go ahead and start the video? So the other thing is, so if we use these surface electrodes, which can be done either in the epilepsy monitoring unit or actually in patients who are being just implanted for this, and what we are doing with those surface electrodes, and this is where we hope that that video is going to decide to run. We got it to run earlier. So what we are doing is we're primarily looking at signals that are in the 50 to 100 hertz. And in the ECOT patients, or these patients have electrodes, what they are doing is they are imagining a particular motion. And what we are looking at is for modulation in this region based on a particular motion. So they might be imagining moving their thumb, or they might be imagining moving their wrist. And we then use those to translate for cursor movement. So now you're going to see is when it's quiet, the ball will drop down. When the subject is doing making a different thought, you'll actually see positive activity, and that is how it will drive up. All right. So this is in this particular video, we have this set up at the patient's home, so we work both in the lab and at home. For these patients, these are 28-day implants. We work with these patients 6 days a week. We typically do 4 to 6 hour sessions. And so what he was doing there, we were using a 3D television. And so he is now doing, these are 3D tasks. And so you can see the target lights up, and then he is driving the ball. This is 100% brain control. So there's no computer assist for this. And he actually was able to learn this very quickly. And by the time that he had finished at 28 days, his accuracy in 3D tasks was at about 80%. He had asked us that the one thing he wanted to be able to do was to reach out and touch his daughter. We just had the robotic arm in the lab at that time, and so this is a non-controlled study in that we just hooked him up and said, hey, try to see if you can move it. And so he was able to generate enough motion that he was able to reach out to somebody. In this case, we also worked with another patient. This patient had ALS. He had loss of the use of both upper extremities, but not his lower extremities. And you can see there, those are the leads coming out on the chest. And with him, we spent a fair amount of time also working with 3D control. This is a different robotic arm. This is actually the DECA arm, which I think some people may have heard of, which is sort of considered the Cadillac of prosthetic arms right now. Michael was not able to generate greater than about 60% success on these robotic arm tasks. We did a lot of work with him where we gave him a little bit of computer assist, so sort of think of it as training wheels. So if they started to drift too far away from the target, there's sort of a limitation. It helps drive them back. But again, he was able to learn this fairly quickly, and being able with cursor control, again, to sort of generate that 80% success within that 28-day window. Just to give you an idea of the learning, this was from one of our subjects. And so you can see we started out with a 2D, 4D task, and the red lines show the amount of computer assist, which we got rid of, and then he actually improved quite a bit after we got rid of it. We then switched to 8 targets, and again, we see him improve. And then this was some days off, and then we switched over to 3D, and of course his success drops off terribly when we first switched to this 3D, but then he's again very quickly able to master that. And that's very consistent with what we saw with all 3 of the subjects who have done work with the ECOT study. Our last ECOT patient was actually probably the most interesting. We went with a slightly larger grid, and we intentionally laid the grid across the central sulcus. So these blue electrodes are actually motor, and then the yellow electrodes back here are actually over sensory. The publications from this will be coming out shortly. One thing that is very interesting is that we found when we were doing motor control, we actually used equally the electrodes overlying sensory as well as motor, and so there's a good indication that you can use signals from both. Because of some of our targeting, one of our first subjects, most of his electrodes were over sensory, and he had very good control. So this is useful when trying to think about, is this something that we can use in patients that have had strokes and may have a cortical defect? From the standpoint of what kind of sensation we had several clusters. So we had one small area, this is over here, where he had sensation just on his pinky. Then we had one area where we had the pinky and the ring finger one where we had just the fingers, so that was sort of this larger area here. Then we had all 5 fingers, and that sort of corresponds to these areas. Interestingly, he also had some sensation for some of these across his inner arm. This particular patient had a spinal cord stimulator because he was a brachial plexus injury. And he commented on the fact that the sensation that he had from the cortical stimulation was not the same as the sensation he had from his cortical stimulator. And a lot of people have proposed that if we do direct stimulation of the cortex, that we're just going to generate these unnatural sensations like the buzz that they get with their spinal cord stimulators. But he very clearly said that these were different, and I think that was useful. He never really characterized them as natural. But I think part of that is that we were using very simplistic stimulation parameters, and probably as we get better at this, we can begin to get closer to some naturalistic movements. Interestingly, he also had a few sensations which produced the sensation of motion. So it makes me encouraged that there is the possibility that we might be able to actually reproduce the sense of proprioception. Talk a little bit about the penetrating electrodes that we've used. These are the standard penetrating electrodes. We're using shanks that are 1.5 millimeters in length. There's a long history with these electrodes. They've been around certainly since the early 90s. And these have been done. There's been lots of work in animal studies. And there are a number of institutions that are using them for human studies. One of the concerns has been that these are unreliable and that they don't last for a long time. If you look really closely at the animal literature, a lot of the failures are actually due to monkeys grabbing their pedestals and trying to yank them out, or sticking their fingers into their wounds and pulling their wires out. Fortunately, none of our human subjects have tried to do that. We did have one husband who tried to pick his wife up by her pedestals. We encouraged him not to do that. This is an example of some FMRI data, and then you can see here along motor cortex that we have placed these within areas that we think are going to be hand and arm. Let's see, for you all, this is not particularly interesting. This is just using an impactor to implant the electrodes. And we do this with a team approach. We have a team in the OR so that if we get in the OR and there's a vessel where we want to put the electrode, we have 2 or 3 subsequent plans so that we can adjust based on the anatomy. Here you can see these are the pedestals now attached. The craniotomy is closed up. The goal is to run the wires away from it. Now, we had a little bit of, and this has been discussed by other people, we had a little bit of retraction around the pedestals with our original surgery. In our subsequent surgeries, we've actually used stab wounds to bring the pedestals up through that, and that seems to have eliminated our retraction. The other thing that is interesting, our first subject was a female with an incredibly thin scalp. Our second subject is a male who's much younger, who's got almost like a Shar-Pei scalp. It's really thick. I'm sure that that is helping. Of all crazy things, the plastic surgeons are really interested in injecting fat stem cells into the scalp to try to help thicken it up in patients who have scar. Someday that might actually be helpful for people with chronic implants, whether it's going to be RNS systems for epilepsy or other things where there's a long-term implant that is thinning the scalp. Part of our surgical team. Just so with these electrodes, we actually are recording single units. And they are quite stable. On average, so the red dots were our original subject. She typically had somewhere around 1.5 units per electrode. Our current subject, we've been a little bit higher. He's tended to stay a little bit higher, around 2. Both of these subjects had implants that were in place for greater than 2 years. So what good is this? We've been able to put an implant into someone's head. Can we do anything useful? And just to show that we could make a robotic arm move was not enough for us. So we went to occupational therapy, and we worked with them with some of the tools they use for screening stroke rehab patients. And so the one we used is the ARAT. And if you get a change of greater than 6 points, that's considered clinically significant from the standpoint of benefit with your rehab program. So our patients, because they can't use their arms, I'll start out at 0. So I'm going to give you a look at here. Well, there it goes. All right, so this is one of our subjects, and what she is doing, this is 7 degrees control, so she can reach in 3 directions, she can open and close the hand, and she has yaw, pitch, and roll of the wrist, so those are the 7. And what we're having her do is simply pick up these objects, and I think what you, now here, this is a nice one because she actually corrects and manages to just get it in the right place. The occupational therapists love these videos because they see that they use the same kinds of techniques to compensate to pick up the objects as you see in somebody who is recovering from a stroke. So they'll do sort of scooping maneuvers to grab things. With the rock, you know, there's this tendency to really grab it so that you can't drop it. But you can see that they can easily grab a variety of object sizes, weights, and densities. So I talked about the fact that we can generate a ton of information and data from these subjects. So we wanted to know, well, how much can you control? So we went ahead and we went to see if we could do 10 degrees of freedom. So now what we've added is we've added a bunch of different hand postures, and it turns out that playing rock, paper, scissors was a great way to practice those different hand positions. Now, it turns out the researcher hiding here in the corner is Andy Schwartz. He'd never played rock, paper, scissors, which is really funny, which just makes him a geeky scientist. So Andy is somebody who I met when I went to my first national meeting. I went to the CNS meeting in San Diego, and I ran into some friends who had been residents at Vanderbilt when I was there. And one of them literally made me call Andy while I was standing there, because he thought we had similar ideas. And I actually rented a car, which I did not have the money to do, and I drove out to Andy's lab in La Jolla. And the first question he asked me was, how do we take the work I'm doing in monkeys and get them into humans? I told him he needed a crazy neurosurgeon, and I continued to tell him that until he was here in Pittsburgh, and we had our first meetings about how we were going to move from an institution that had nothing but animal studies to being able to do human studies. Mark mentioned the collaboration with CMU. So one of the things that we have done is we have worked with the robotics community at CMU, and we have looked at coupling robotics, and in this case this was a visually assisted robot, with human control and blending the control. And what I'm going to show you, so the red bars were performance from our 2 subjects on a task. And then what we did is we blended their control of the robotic arm with the visually assisted control. So the visually assisted control is the robot identifies an object and it just goes and grabs it. And you can see that they were then able to begin to perform these tasks much faster. And when they look at the perceived difficulty, you see that with the robot assistance, the perceived difficulty becomes much easier. So to give you an idea of what this looks like, all right, let's get started. On this side is shared control, and the task is to grab the wooden block. This is same time, right, but no shared control. And you can see that it's much more difficult to grab the block. This is the other subject who also participated this. And again, you can see that with that shared control, and one of the things that the subjects liked about this is they made the decision to reach to the object, and the robot didn't take over until they were approaching the object. And if they were approaching the wrong object and they gave this arm the signal to pull back, the robot stopped and waited until they approached the object again before it initiated the graph sequence. So we did sensory with the cortical ECOT electrodes, but we've now also done these with penetrating electrodes. And so we can see here, these are our 2 motor electrodes. Here are our electrodes in sensory cortex. You'll notice they're oriented at different directions. We were very specifically trying to get a broad sense of the hand, or as much of the hand as we could, and so part of this was trying to target specific areas. This is the example of some of the MEG data. In this particular case, we were stimulating, trying to see if they had a response. This patient is an Asia B C5 quad, so he does have some sensation. So these are the color coding here along these grids show where it corresponds to the hand. So you see we were able to achieve responses all the way across the bottom of the fingers and then a little bit out on the pinky and a little bit out on the index finger. What is interesting about this is that he responded that either it felt like it was surface or just below the surface. He classified probably about 10% of these signals as being natural, and all the rest of them as maybe natural or close to natural. He never had anything that was painful. He never described anything as being a buzz or something we might think of as akin to that spinal cord stimulator sensation. And so that was really interesting. The other thing that we thought was great was if you look at the psychometric or the data from animals, you would normally get a detection curve that are these nice sinusoidal curve or sigmoidal curves. We see the same thing with our human study. We saw that, in fact, the thresholds for these decreased over time. So these have been really very robust, and we've seen no change in our ability to record from these electrodes even after stimulation. So we're not damaging the tissue around them when we do this. Now, I'm afraid the audio is not going to play. Is there any way to turn up the audio? Well, it's in HDMI, so it should. All right, so we're going to miss out on this. So what we're doing here is you can see here on the inset, this is Rob Gaunt. He is one of the two PhDs in our big joint lab who's really been focusing on sensory. And he's pushing each of these fingers. And the reason we're using this video is because he gets it right 20 out of 20 times. He wasn't always that good, but he was about 90% correct with these stimulations, as long as our stimulation threshold was above the 50% detection threshold. So this is really cool. Now not only can we control an arm, we can generate sensation. And so the reason we can do this with this particular arm is there are sensors in these fingers that actually we're using the torque sensor, so they're detecting motion. We've now started doing things with him, having him grab objects and determine whether they're hard or soft objects. And so he's actually getting pretty good at that. But how do these are sort of preliminary studies anyway, so the question is, how do we prove that this is going to help him do motor tasks? And so what we're working on is developing motor tasks that are complicated enough, but not so complicated that they're hard to make them work, to see if we can start to determine whether having this sensory feedback actually allows him to improve his ability to do these tasks. This is, and I think many people have brought this up, this is a huge collaborative effort, not only from the standpoint of all the different types of work that goes into it, whether we're doing animal work, working with our target populations, because it's hard to find people to volunteer for these studies. We've done work with the epilepsy unit in the past, and we'll continue to do that. And then we've been using both single unit, ECOG, the sensory integration that we're now doing, plus the MEG. Many institutions pick one and focus on it. And so we've been sort of unique in the fact that we have spread ourselves across all these, but we feel that each one of these feeds into the other to really kind of help us be a stronger program. The other thing is that it's great for the intellectual debate as to is less more or is more more as we continue to work forward with this. As I mentioned, the many people who have helped is more than I could list. We have had funding through DARPA, NIH, the Department of Defense, NSF. We've had lots of institutional help. We have collaborated with Johns Hopkins primarily outside of Pittsburgh at this point, although we're looking at some more future stuff. And certainly we can't do this without the volunteers. Thank you.